Recording in progress.
Good day, everyone, and welcome to the last session of Seminar 7 of LPHYS'23. I'll be the chairman of the session. My name is Fabrizio Cecchettini. I'm from INRIM in Italy. I suggest, since there are just 2 talks today, to wait other, let's say, 5 minutes or so to see whether some further people joins us. And I will give each speaker will have half an hour, including question time. So I will just warn 5 minutes before the time ends, or no, let's say 10 minutes before the end of the full time of the presentation, so that the speaker can decide how much question time should be left and how much time should be instead dedicated to the conclusions of the talk. Okay, I think one of the speakers is already here. Is it okay for you, Elena? And you're muted,
yes,
so
perfectly.
Okay,
Perfectly fine for me. Yes.
so it's let's wait until 2:05 or even 2:10 if people.
Yeah, no problem. I would be happy if someone will join.
Yeah, sure. So it's okay. I think the second speaker is not here yet, but okay. Probably the second speaker of the session just joined, although I'm not that good in Cyrillic. I think it's Polina Vinitskaya, what I can read from the. Then what it was. Okay. So we'll wait another couple of minutes, and then we start with the first talk. As I already said to the first speaker, both speakers will have 30 minutes, including question time. I will give a 10 minutes warning just before that, 10 minutes before the end of the time slot, so that the speaker can decide how much time has to be dedicated to question time and how much time to the conclusions of the presentation. So I think it's time to begin the session. And to do that, let me introduce the first speaker, that is Dr. Elena Losero from the National Institute of Meteorological Research in Torino, Italy. And she will give a talk titled Biosensing with Nanodiamonds, Detection of Temperature Variations in Neurons using Nitrogen Vacancy Sensors. So Elena, the floor is yours.
Okay, perfect. So I will share my screen. Let me do that. Okay, then. Okay, can you see the slides in full screen?
Sure, perfectly.
Okay, great. So good afternoon to everybody. Thanks for attending my presentation. So as Fabrizio introduced, I'm Elena Lozero, and I'm giving this talk about biosensing with nitrogen vacancy center today. I'm working at INRIM, in particular in the quantum optics group. Okay. The quantum optics group at INRIM, led by Marco Genovese. Our group is working on different aspects of quantum optics, like quantum imaging, metrology and sensing, metrology for quantum communication, foundation of quantum mechanics, and color centers in diamond. In this talk, I will focus on this aspect in particular to show you how color centers in diamond, and in particular NV center, can be used for sensing and biosensing. So let me tell you something about what NV centers are. Maybe some of you are already familiar with them, but I prefer to do a brief introduction to these defects. So diamond is made of carbon, as you all know, but there can be defects into the lattice. In particular, there can be NV centers, which are created by a nitrogen atom and a vacancy nearby. These centers can be in different charge states, which are called NV plus, 0 or minus, depending on the charge of the state. And for sensing, the centers which are particularly useful and then can be exploited are the NV minus centers. These centers are particularly interesting and quite well explored nowadays for different applications, like computation and sensing, as I will tell you better. So this NV center can be excited with a green laser in a non-resonant way. So you don't really need a super good lasers in terms of light width. You can use a normal laser in the green region of the spectrum, and you will obtain some fluorescent light from these centers with a 0 photon line and 637 nanometers, so in the red region of the spectrum. The spin state, the ground state, is a spin triplet state with ms equal 1, +1, -1, and 0. If you don't have any magnetic field in your environment, the +1 and -1 states are degenerate, and are anyway split with respect to the ms equal to 0 state by a certain amount, which is called the zero field splitting, which is in the microwave range. The most important property of these centers is that their photoluminescence is spin dependent. This means, as is schematically reported here in this diagram on the right, that if your center is in the ms equal to 0 state, and you excite it and then collect the fluorescence, what you have is a high level of fluorescence. On the other case, if your state starts from ms equal to plus or minus 1 and then you excite it to the excited state, in this case, the level of fluorescence is lower. So this is a way to distinguish if your state was initially in the ms equal to 0 or in the ms equal to plus minus 1 state. And using microwave, you can manipulate the initial spin state, and therefore moving your state from ms equal to 0 to ms equal to plus minus 1 if you match the resonance frequency of the zero field splitting, so this DGS value. The different photoluminescence value is due to the fact that when your defect is in the ms equal to plus or minus 1 state, the excitation can pass through an intersystem crossing mechanism. And therefore, instead of emitting light like in the ms equal to 0 state, you can pass through these metastable states, and therefore not emitting any light in the red region of the spectrum. So based on these electronic structures and these properties, you can exploit these centers for sensing, in particular for the fact that the electronic level structures that I present here depends from the environment where the NV center is. This means, for example, then that if you have a magnetic field at the NV center position, the position of the ms equal to plus and minus level will change, and therefore the DGS value will, the distance between the ms equal to 0 and ms equal to plus minus 1 states will change accordingly. So the idea of NV center for sensing is summarized in this slide. So what we have typically is that we have NV center in our diamond. We shine a green laser on the diamond and on the NV center which are in it. And at the same time, we send microwaves and we collect the fluorescence light. We sweep the microwave around the zero field splitting value, and we collect the fluorescence. Once the microwave frequency match the resonant frequency, which is the distance between the ms equal to 0 and plus minus 1 states, what we have is a lowering of the fluorescence, because in that case, we are probing the transition between ms equal to plus minus 1 and the excited state, rather than the one from ms equal to 0. The interesting thing is that if the electric field, the magnetic field, or the temperature is changes at the NV center position, this will result in a shift of the resonance frequency. And therefore, if we are acquiring this photoluminescence versus frequency, we will observe a shift of the position of the dip. And from this position, we can infer, for example, the magnetic field, the temperature, or the electric field at the NV center position. Okay. As I said, NV center are sensitive to electric field, magnetic field, and temperature. Magnetic field is one of the most typical application of NV sensing. There are many papers which explore this option. Electric field is also quite interesting, but it's a bit more challenging because the coupling with NV center is lower respect to magnetic field, and therefore is still explored a lot, but there are not a lot of real application, let's say, for the moment. In our group, we are mainly focusing on temperature. Because also in this case, because NV centers are sensitive to temperature as well. And you can somehow imagine this fact, considering really that this splitting here, the splitting between the states ms equal to 0 and ms equal to plus minus 1 depends from the interaction between the spins in the orbital structure. And therefore, if temperature change, also the crystal lattice will change, and in particular the distance will increase for a temperature increase, and the interaction will decrease. And this will lead to a modification in the level structure that we can probe with the mechanism I told you before. So here, what I'm presenting is what can be done for temperature sensing. So the idea is that if we work in the idea is that for small temperature change, we can assume a linear regime, and therefore we can assume that increasing the temperature, also the position of this dip will change linearly with temperature. So the plot that you see below in the center is an experimental acquisition that we made in our lab. So what we report here is this photoluminescence versus frequency, which is called ODMR spectrum, for two different temperatures that we control with a thermocouple on a bulk diamond chip. So we have our diamond, we measure the ODMR for 28 degrees, then we change the temperature and we see what happens for 38 degrees. And as you can see, the spectrum is shifted. And this shift can be, from this shift, you can find the temperature shift which induce it. One important thing that I didn't mention clearly probably is that with the NV center, you can measure temperature shift, but you cannot really measure the absolute temperature. So this is something which is important and that we need to keep in mind all the time. So with we can easily measure temperature shift only, but not the absolute value. Okay, so this is our sensing. This is our sensor. But now, why it is interesting in the biological context? So why do we want to apply NV? Why do we want to use NV centers and thermometry in particular in biology? So the idea is that temperature can be extremely interesting to be studied in biology and in cells, because it's something that with current methods is still not really explored, and it's still not really clear what's happening in terms of temperature at the subcellular level. Because the tools that we have to measure temperature typically measure temperature at a much bigger scale. So maybe we can have the measure of temperature of the entire bath, of the entire culture of cells, but it's much more difficult to say something about what is happening at the cellular level, and even more difficult to assess something about what is happening inside the single cell. So this is a bit our goal, is to explore what's happening inside a single cell, and this technique will allow us to do that. NV centers are particularly interesting, more in general, in the biological context, because they can be operated at room temperature. This is, of course, extremely important because cells are typically, are extremely sensitive to temperature. You really need to to maintain them at a specific temperature in order that they can be healthy. And then also diamond is biocompatible. This is also important because this is why our technique is really biocompatible and can be not destructive. So this feature of NV center are at the basis of the use of NV center in different bio applications. In our case, we use ODMR techniques which is the one that I discussed before. There are also present in literature other techniques which use NV center in diamond, but from a different perspective, like for example measuring T1, which is another properties of NV centers, but which is not is not what we are exploiting in this work. So there are different options that you can have if you want to explore diamonds for temperature sensing in cells. One option is to use bulk diamond. With bulk diamond, I typically mean diamond chips, which are macroscopic, something like typically you can buy bulk diamonds of 3 millimeter by 3 millimeter by hundreds of micrometer thickness. And what you can do with those chips is to grow cells directly on your chip. And then eventually you can also nanostructure the chip in order to improve the adhesion, to improve the collection efficiency of the fluorescence light, and these kind of things. This approach was the approach that I had that they follow in one previous work, which was done at EPFL in Switzerland, and which I report here in one of the images that really show what we were doing in terms of culturing cells onto a nanostructured diamond chip. So what you see here are neurons, and all these small pillars are nanopillars of diameter of 500 nanometer and height of something more than 1 micrometer. So this is a possible approach. There is also another approach, which is the one that we are actually having here at INRIM, which is to use nanodiamonds. So nanodiamonds are, of course, nanodiamond are particles made of diamond. In our case, we use sizes of around 200 nanometer. There are also smaller nanodiamonds, but for us, it's important that they are big enough to collect enough signal. And if the nanodiamonds are too small, they contain not enough NV center and the signal is very low. And then what we do with these nanodiamonds is to internalize them into cells. For doing that, it's quite easy in the sense that you need to put the nanodiamonds into a solution and then the cells in the same solution, wait for a while and wait that these nanodiamonds are really internalized into the cells. Then you can really see them at the optical microscope because they are fluorescent, as you can see in this image on the right. So once they are internalized, you can use them as I told you before. So you can really focus on one nanodiamond and extract signal from it. So I was introducing that we want to measure temperature. And here I summarize a little bit why we are interested in temperature. Temperature is interesting in the biological context because regulates the speed of ion channel opening in cells and pattern activity of firing neurons. In our work, we are particularly studying neurons. Also other cells are possible, but we are focusing on neurons for the moment. And then temperature is also important because intracellular temperature is affected by a variety of biochemical reactions, which occurs during cell activity. And finally, temperature may be altered under pathological conditions. So in perspective, measuring temperature with a high degree of control can be extremely important to monitor the diseases and eventually find some clinical solution. So for measuring temperature, nanodiamonds are particularly good because they are biocompatible, insensitive to biological environment, and they are also small enough to assure a very high spatial resolution below the cell dimension. L for a lot. The risk that you have while working with nanodiamonds and diamond in general and NV center is that the sensing protocol that you use implies laser and microwave irradiation on the sample. And here we are talking about biological samples, which are sensitive to these things, to both laser and microwave. And therefore, both lasers and microwave needs to be limited as much as possible. And this is something that we try to do with our sensing protocol. So our goal, the goal of the work that I'm presenting here, was really to modulate, to understand if a modulation of neural activity was correlated with intracellular temperature variations. For answering these questions, we can consider three different groups of cells. One control case, where there was simply neurons into a saline thyroid solution, which is the typical liquid where they live in cultures. Then we add a substance which is called picrotoxin, which is known to enhance the firing activity of neurons. So this is one case where that we want to compare to the control case to see if the temperature was different or not. And then we also add another substance, which is called tetrodotoxin and cadmium chloride, which are known to drastically lowering the neuronal firing activity. So these are the three cases that we wanted to compare in terms of temperature variation respect to the control case. The experimental setup that we used is schematically reported here. So we use a confocal microscope with a microwave control for ODMR acquisition. So you see here we have a laser excitation path in the green region of the spectrum, 532 nanometer laser. The power was kept low enough not to damage the sample. In particular, our power was below 1 milliwatt at the sample. The laser was focused on the sample here, and the stage was adjusted in order to properly host the neuronal culture. So there was a temperature chamber to guarantee a stable temperature of the bath, and a petri dish, which is the typical plate that are used in neuroscience for culturing neurons. And then below this, below the neuronal culture, there was an antenna to send microwave to the sample. The same objective, which was used to excite with green laser the NV center into the nanodiamonds, was also used to collect the light coming out from the nanodiamonds, which was separated from the green light using a dichroic beam splitter. And then the red light was sent to both a single photon detector for the real measurement, or to a CCD camera to have a wider image of the sample, and therefore to find the nanodiamonds in the preliminary step of the process. So the idea was
10 minutes left.
Okay, great, thanks. So the idea was first to, of course, find the nanodiamonds, find the cells using a wide field CCD camera, and then focus on the nanodiamonds of interest for acquiring the spectrum. So this is the setup that we developed at INRIM for measuring, for acquiring the signal from the nanodiamonds and assess a temperature variation in the different cases. At the same, but for this experiment, we were also using another setup at the neuroscience department for measuring the neural activity independently after the addition of these different substances. And for doing that, we are using MEA, multi-electrode array system, which is something quite standard in neuroscience, where you really measure the electrical activity of the neurons using all these electrodes that you can see here in the image on the left. Okay, so let's move to the sensing protocol, which I already briefly described before. So our sensing protocol was mainly driven by the necessity of keeping as low as possible the sample exposure in terms of light and microwave. So before I was saying, okay, if you want to measure a shift in temperature, what you can do is to acquire this ODMR spectrum, which is the photoluminescence versus frequency, in different moments, and averaging enough in order to have a signal to noise ratio which is good enough. But of course, if you want really to have a very good signal to noise ratio, you need maybe to acquire for a long time, which can be too long for cells. And therefore, we found out that it was better to have a different protocol, which we call here differential ODMR, and where you really limit the exposure time of your sample and the microwave that you send to your sample. For doing that, what you do is to, just briefly, is to acquire a full ODMR spectrum just once. And then you post-process this spectrum in order to have a differential spectrum, which is basically the derivative of the first one, as you can see in the red line here. And then what you do is to acquire the photoluminescence just at few frequencies around the zero, around the linear region, so that you are limited. You can really limit the acquisition time and exposure time of your sample. And then you can recover the variation in temperature, basically you exploiting the linearity of your region, of your working region. So using this sensing protocol, let's go to the results of our work. So what we demonstrate with our technique was that actually there is a significant temperature variation in the three cases. So if we compare temperature of the control case with the case where we added picrotoxin, which means the case where the neurons were more active, the neuronal activity was bigger, then we observe a temperature increase of 1.02 degree. While in the opposite case, that is the case where we added the substances which lowered the neuronal activity, well, in this case, we observe a decrease in temperature of around 0.5 degree. And this result was extremely great and interesting because it was really the first time that nanodiamonds and NV center in diamond in general were used to measure temperature in cells for a temperature shift induced by the cell metabolism, and was not just to measure temperature induced by something else like thermocouple or change in the bath. So this temperature change is something which really comes from the cell's activity, and this is interesting results which can be explored more also from the biological point of view to better understand what's happening in terms of cells metabolism and temperature, which is still not clear and interesting to find, let's say, to explore. So just to briefly mention, then if you're interested, I can tell you something more. Of course, our goal was really to understand if this temperature variation was related to the cell metabolism and not to something else, and therefore we performed several cross-check measurements in order to be sure that what we observed was due to the metabolic activity of neurons. So for example, we measure what was happening in terms of temperature variation just on the nanodiamonds which were not in cells, for example. And we observed that in this case, there were no temperature variation at all. So this is a good validation of our results. And so in conclusion, what we did in our work was to use nanodiamonds internalized into cells and neurons in particular to assess that different firing mechanism of neurons induce different temperature in the cells. At the moment, the sensitivity of our setup was already enough to clearly observe this phenomena, but we estimate that sensitivity can be improved about a factor 3, simply improving the photoluminescence collection of our setup implementing nanodiamond tracking, which means really to follow the nanodiamonds that maybe can float into the cells, and also to have more better nanodiamonds, in the sense that nanodiamonds with more NV centers, which means more signal, basically. And just last slide for giving you some outlook of our research that is going on INGRIM nowadays. So what we want to do is to keep working in this direction, also collaborating with other neuroscience, either neuroscientists and biologists in general, to monitor temperature and in different cells, for example, for different cells and different biological processes, to use this technique to other contexts. Then what we want to also do is to use functionalized nanodiamonds to measure temperature in different locations inside the cells. Because at the moment, we know that nanodiamonds are inside the cell, but we are not sure about where they are exactly, or better, they can be everywhere inside the cell. But there are techniques which allow us to functionalize the nanodiamond surface in order that they attach to specific parts of the cell. And this would be extremely interesting to map the temperature inside the cells and see, for example, if there are specific gradients between different parts of the cells, or if there is some part of the cells which heat up more than others. So there is space for investigation, let's say. And finally, what we are also working actively on these days is to monitor temperature and electrical activity with multi-electrode array at the same time. In the work that I presented, those two things were done separately in two different moments, but it would be nice to have all these measurement tools together in order to have a multi-approach measurement of this kind. And I think, that I'm also running out of time, so I will thank you for your attention, and I'm open to any question if you have.
Okay, thank you, Elena, for the very nice presentation. And we can take one or two questions, I think. So, is there any question? Yeah. Well, there is one question from Dmitry Kuprianov. So, please.
Yeah, thank you for your talk. It's very interesting and helpful for people, I believe. But I have a technical question.
Yes.
How many atoms, carbon atoms, are in your nanodiamond? And if you take this number, you take root of it, we can estimate the fluctuation of the temperature as a thermodynamical parameter. And maybe it's comparable with the value of temperature variation inside the biological object you studied.
Okay. I'm not sure I understood the question. I mean, these nanodiamonds, you can...
If I temperature, normal room temperature by root of number of carbon atoms inside your nanodiamond, I estimate the internal thermal uncertainty for temperature for this macroscopic object.
Okay. I mean, we to estimate the number of atoms, you can assume these nanodiamonds as sphere of 200 nanometer of diameter, I think. So if you want to do this calculation, I think it's something that can be done. What I can tell you is that in our work here, we did many test measurements to really check that every temperature variation that we observed was somehow real. So, for example, before measuring nanodiamonds in cells, we did the same measurements outside of the cells. So, just using many nanodiamonds in a bath, so not in the cells, but just in the solution, to see if there was a dispersion of temperature, which, I mean, if the dispersion of temperature was not too big. So, if we could really see the same, if we can extract the same values from all the nanodiamonds, and this was the case. So we checked that change in temperature, we could actually observe a change in temperature.
Yeah, I think you are right, but I just advise you to make this estimation. It would be useful. Thank you.
Yes. No, okay. Yeah, I will note it down. Thanks. Yeah.
Okay, I think we have to proceed with the presentation. So thanks, Elena, again.
Yeah. You're welcome.
Keep going with the last talk of the session, actually. That is by Dr. Polina Vinetskaya. You're muted, so you have to unmute yourself.
Hello, dear colleagues.
Okay.
I'm actually...
And she's from Lomonosov Moscow State University, and she will deliver a talk titled Experimental Implementation of Active Basis Choice for QKD with Entangled States. So, Polina, the floor is yours. I will just give you a 10 minutes warning before your time expires. So you can decide the amount of questions you want to take.
Thank you very much. Hello again, dear colleagues. My name is Polina, and sorry, I'm actually a student, not a doctor. And today I'll tell you about our
soon-to-be doctor.
Our work on implementing the BBM92 quantum key distribution protocol with the active basis choice. The two photon states are generated as a result of spontaneous down conversion in a nonlinear crystal. The Bell states required for the implementation of the protocol are obtained as a result of the interference in the Sagnac interferometer. Such a scheme is quite stable, and it is widely used as an entangled state generator. In this figure, you can see the complete Bell state generation scheme. The source at a wavelength of 780 nanometers generates a continuous radiation with a power of up to 30 milliwatts. To achieve the maximum efficiency of photon pair generation in the crystal, the pump beam waist should coincide with the center of the crystal, and the relay length should be equal to the length of the crystal. In order to fulfill these conditions, lenses 6 and 7 are put to focus and determine the position and the size, the radius of the pump beam waist. Filter 13, which you can see on the picture, are used to filter the signal from the pump power. Thus, only a signal of a wavelength of 15 and 50 nanometers enters the collimators 14. This slide shows the results of measuring the single counts rate recorded from the collimators 14 from the previous slide using indium gallium arsenide single photon detectors. Unfortunately, at generation frequencies on the order of 10 kilohertz, here, the detectors go from the linear mode, depending on the pump power, to the saturation mode. This is due to the fact that the frequency of registration cannot exceed the reciprocal of the dead time of the detector, which is 15 microseconds. Also, dark noises have an effect. In any case, measurements at the maximum power of the pump laser cannot be effectively carried out with these detectors. So the graph shows the data for different attenuation values. Further, the average values found under absorption of a linear regime will be given. Here, you can see the results of the measurement of coincidence counts rates, also for different attenuation values. Since this value depends quadratically on the pump power, the coincidence frequency at the maximum power of the pump laser is actually much higher than actually measured. As you can see, it is actually in order of 2.6 kilohertz. The next step in the implementation of the protocol was setting up of Alice and Bob's receivers. The original protocol proposes the use of passive basis choice, as you can see here, which uses symmetrical beam splitters to choose the basis. We use a more practical one, an active basis choice, which uses the electro-optical effect of a nonlinear crystal to modulate the polarization and to choose the basis. Also, as one basis, we use the original vertical horizontal as in original scheme, and it corresponds to 0 voltage applied to the crystal. As the second basis, instead of the diagonal one, as in original scheme, we use circular polarized R and L states, which are also orthogonal to the first basis. The corresponding phase shift voltage is written on the slide, and it is equal to 710 volts. It can be proved that these two schemes shown on the slide are equivalent in terms of the protocol BBM92. The visibility of the interference in the Bell state, in the Bell test, was also measured. The polarization controllers were adjusted to the position at which the maximum and minimum of joint responses of the Alice and Bob detectors was observed at 0 voltage on electro-optical modulator. Visibility was 91.4%. With the help of polarization modulators, quantum bit error rate was found for matching and non-matching bases. Rejecting those measurements that do not fit the matching condition, I mean matched basis or non-matching basis, the percentage of errors was found to be 7.89% for matching basis and 40.62% for non-matching ones. Also, the rate of quantum key generation was measured. It was, as you can see, 560 bits per second. The result, BBM92 protocol was implemented with active basis choice at a frequency of 15 and 50 nanometers. The dependence of single and coincidence counts rates on the pump laser were measured, and assuming a linear mode, the quantum key generation rate was found to be 0.5 kilobits per second. Also, quantum bit error rate values were found. It was, as I said, 7.89% for matched bases and 40.62% for non-matched ones. Thank you for your attention. I'm ready to answer your questions.
So thanks, Paulina, for the nice presentation. And you've left basically 20 minutes for questions. So I hope there are a lot of them. Any question?
I have a few questions.
Okay.
Can you hear me?
Yes.
Yeah, sure.
Thank you for your talk. The first question is what type of nonlinear process you used to generate entangled states?
We used, as I remember, sir, I'll send you more information by the mail. I don't remember the name.
Okay. Could you please name the crystal which you used to generate these entangled states?
It's a type 2 down conversion and PPKTP crystal.
Oh, okay. And the third question is why you used or you're using 1.5 micrometer or 1550 nanometer instead of 1 micrometer? Any reason?
We used
this frequency. Why this frequency?
We used telecom frequencies, and also we have tried to make the same scheme with visible frequencies, and it was quite good, but it was more interesting for us. So I presented just this part.
Mm-hmm. In your previous slide, I have seen free space. So are you also going to use this kind of setup for free space, or with fiber you are planning?
Can you please repeat?
In your previous slide, I have seen a free space. So the citation in the bottom is a free space. So question is, do you plan to use it also for the free space?
We used this in our setup, as I said, which was on the visible spectrum. And here I put this citation to cite to this active base choice scheme, because they used it with fiber optics, this part.
Mm-hmm. Thank you.
Thank you.
Other questions?
Maybe one more question, sorry.
Yeah, sure.
Why BBM 92? Why not E91 protocol?
We were interested in this setup, and it's just the choice, as I think.
Mm-hmm. Okay, thank you.
Thank you.
Again, other questions? Well, I have a couple if there are no other questions. So just curiosity, because the setup is interesting. I mean, about the telecom Sagnac interferometer that you built up, do you remember some of the technicalities, or you were just focused on the protocol? Because I have one question about the Bell inequality violation. Which is the value that you obtained in the Bell inequality violation that you? I mean, I see the visibility for the Bell test is 92%, 91.5% almost, but I couldn't see any Bell inequality, Bell parameter value that you gave with your setup. Maybe I missed it.
Oh, as I know, we haven't measured that. We plan to do this in the future, but at this point,
it's written.
Focused.
What you write here in this slide is visibility of the interference in the Bell test. So you didn't perform the Bell test.
We did it by adjusting the polarizators, and we compared the maximum and the minimum value.
That's the visibility. That's how visibility is computed.
Yeah.
But I mean, so you this is the visibility for the forthcoming Bell test. You didn't actually perform the Bell test.
As I know, no.
Okay.
We are planning to do this, but
value.
At this point we focused on the protocol itself, and we had to measure this just to be sure that everything is okay and we can go further.
Okay, and other couple of questions that I have. How robust is your source, and in general your setup, to noise caused by temperature variations or fluctuations due to vibrations or whatsoever? How good does your entangled state remains in time, essentially?
It's quite robust. We, as I know, we didn't measure it like with the graphs, but it is quite robust by temperature. And for another scheme, it was quite obvious by measures. But here we had some problems, as I said, with detectors. So we couldn't measure it very, how to say, clearly. But now we are able to make same measurements with another better detectors. So we are going to measure it too.
Okay, and just last question by my side. Which is the next step? I mean, you're trying to do something else, or I mean, you're trying to improve this setup and to do some further experiments, or if there is something that you can disclose, of course.
Of course, we are going to improve this setup, and there is like the most obvious problem is the problem of the detectors, the saturation mode, and now we are able to make the same measurements with another detectors. So we will be able to measure more parameters, and also we will be able to improve the scheme itself to improve other already measured parameters to be higher.
Because from the numbers that you gave before, I think that your detectors are indium gallium arsenide, right?
Yeah.
Pre-running ones. Probably a solution would be going to SNSPDs. There are superconducting detectors that would definitely are much better in terms of both quantum efficiency and dark count rate, and even that time. I mean, they require a cryostat, so they're not that easy to use. But
thank you very much. I'll think about it. We are going to use another indium gallium arsenide detectors, but they have much better values of efficiency.
Probably the one with Stirling motor that lower dark count by lowering the temperature so much.
Yes.
Thank you for your nice presentation and for a lot of questions that we decided to take. And if there are no other questions, I think
I think I have a comment, not really a question. BBM 92, right? Protocol has been known to be less vulnerable to attacks and also to be able to be transmitted over longer distance. I think this is the point that Paulina did highlight, is less vulnerable to some of the imperfections. So I think that is something you could highlight.
Thank you very much.
At least compared to BB84.
Okay. BB84, no? Right?
Yeah, compared to BB84, I think BBM 92 has some advantages.
So it is most robust to the losses or depolarization loss, or what? Or polarization?
For one thing, of course, it only used 2 states, right, instead of 4. So it's of course not immune to all sorts of attacks. It's still subject to that, but it is generally considered to exhibit fewer vulnerabilities compared to BB84.
Because in fiber, loss may be photon loss, and then it may be polarization, depolarization, or phase degradation. So I think, or maybe scattering, the third type of loss mechanisms. So, but it's good that you have shared this information that BBM 92 is more robust to
BB84.
Yeah.
At least.
Okay.
And also longer distance. I think that was shown many years ago by Watts, at least theoretically, and Yamamoto.
But could you please explain what is the the difference between E91 and because E91, if I understood correctly, that protocol is using entangled state, and here also entangled. So the difference between them, if you know.
I think, yeah, Paulina should be able to
Sorry to
explain.
interrupt you.
Yeah.
As I know, the differences in number of states and bases.
Number of bases. Okay. We can represent in diagonal basis, anti-diagonal, and horizontal and vertical, right? What you are saying.
Yeah, here, BBM92 is pretty equal to the BB84, but using the entangled pairs. So we use
E91 requires 3 bases per both Alice and Bob, and probably, if I remember correctly, BBM92 requires 2 bases for both of them, right?
Yeah.
So that it should be, this should allow for an improvement on the key rate, because essentially in the E91 you actually distribute the keys 2/9 times if you randomly choose the among the 3 bases, because only 2/9 are you guess both Alice and Bob guess the 2 bases of the suited for the communication. Otherwise, in 4/9 cases, you can compute some part of the Bell inequality test, and in 3/9 cases, you don't do anything of this. Basically, that's the how E91 works.
Thank you.
You're welcome. So, I think we can close the discussion and thank all the speakers of the session, all the participants of the session. And with this we close seminar 7. I don't know if anyone else has anything to say. I don't think so. So it's I can close the session for today. Thanks to everyone.
Thank you very much.
Bye.
Bye, friends.